We are delighted to have Dr. Frank Petersen from Novartis, me. And I'm very delighted that we've managed to actually get him to come over to WIPO to give us the benefit of his extensive experience in research. He's the head of natural product research at Novartis in Switzerland, and he has worked extensively on a number of collaborations around the world. And here he's going to give us the experience of what are the key pieces that need to work together to make these collaborations a success, and what are the kind of things that Novartis looks for, not only in partners but also in the policy and legal frameworks. And Frank, over to you.
Thank you very much, Andrew, for your introduction. It's a true pleasure to be today with you here to share some aspects on industrial natural products research. And as Gupta has pointed out, it's a very complex endeavor, and a journey, as you said, Andrew, is a very long one. And patience is of imminent importance when you work in the natural products arena nowadays. So when we take a closer look to that slide here, you see that in average, it takes us about 10 to 15 years to develop in a successful way a drug reaching the market, which is paralyzed by an investment of 1.5 billion. These are the figures which have been published approximately 9 years ago, and I ran through the literature just yesterday and saw multiples of these figures in the meantime, which is really showing and underlying how complex drug discovery is in the pharmaceutical industry. We need millions of compounds which are screened in our HTS facilities to identify the mentioned thousands of active compounds really contributing to this kind of attrition rate you see here, ending up with one successful market introduction. Well, in the natural products arena, from the very beginning, it was a wonderful time. So 200 years ago, every isolated active principle turned out to become eventually a drug. So this holds true for morphine, this holds true for podophyllatoxin and some derivatives of it, and also the reserpine. Or in other words, these compounds stood at the cradle of the pharmaceutical industry from its very beginning. Well, with these kinds of successes, the pharmaceutical industry reached out for biodiversity-driven drug discovery activities. We subdivide our ecosystems in two major parts. The first one is the terrestrial ecosystems characterized by megadiversity regions, hotspots, for example, in the tropical rainforest, where we have approximately 4% of the overland surface, and we see 50% of all species on Earth. On the other side, we have the maybe 20, 30 years ago detected marine ecosystems of relevance for the natural products screening activities. We have here the highest degree of biodiversity, and 90% of all organism classes are here. You should be aware of that from these major ecosystems, we know approximately 150, 170,000 structures which are in public domain, which have been disclosed in scientific publications during this long period of time. When we analyze how many natural products classes have been introduced just during the last 30 years, you see the figure here on the right side. These are approximately 25 new natural products classes reaching the pharmaceutical market according to an analysis of Ganassan from 2008 and complemented by internal studies in my group. I think it's obvious that the pharmaceutical industry cannot live alone from natural products, and this was the reason why further technologies in drug discovery have been implemented in this period of time. At the very beginning, as pointed out in the second slide, natural products have been the only source for chemical diversity. This is different nowadays. We have the synthetics derived from medicinal chemistry, combinatorial libraries. We have witnessed the onset of virtual screenings. We have the recombinant proteins, the antibodies. All these kinds of drug discovery activities are today in the armamentarium of the pharmaceutical industry to identify new drug compounds for the unmet medical needs. And during the last, I would say, 15 years, all these activities have been increased, and this was parallelized by a significant decrease in all the natural products efforts in the pharmaceutical industry in the Western Hemisphere. So when I started my career at Novartis, or at Ciba-Geigy to be more precise, you see more or less a list of the who is who of the pharmaceutical companies involved in pharmaceutical research, starting from natural products. But during the last approximately 15 years, we saw a clear reduction or even termination of those programs, mainly due to the reduction of antibiotic research and the focus on new drug discovery technologies, as pointed out before. We also have had the problem that we have both natural products research no compatibility with high throughput screening concepts. In addition to that, we saw a strong competition between the synthetics and the natural products derived libraries. And finally, also with the onset of the CBD as a positive element, how the originating countries can interact with the Western countries and the Western pharmaceutical industry. We saw here increasing legal uncertainties around IP discussions and the ABS obligations in the CBD content. This led in the last approximately 15 years to almost complete disappearance of the Western pharmaceutical industry dedicated in former years to natural products research. And when you analyze the list on the remaining, or of the remaining groups, you have Pfizer involved with a smaller group in that area. Novartis is the only company with a bioprospecting activity, which is really a considerable one, and the rest of the companies are focused or are concentrated in Japan. However, Japanese research groups never believed in the benefit of biodiversity-driven natural products research, and they did this kind of research within their own country, because Japan is reaching various climatic zones, and you have a high diversity there. So there was no real need to reach out for other biodiversity-rich countries. I would like to give you an introduction and somehow also an overview on our activities where we reach out for bioprospecting partnerships in the Asian area. All these kinds of projects are strictly under the regulation of the CBD, which are also mentioned and defined in the contracts we are signing with our partners in this area. We put here a clear emphasis on sticking to the regulations with regard to protection of the biological diversity, the sustainable use of the leveraged genetic resources, and also we are putting a focus on fair and equitable sharing of the arising benefits. The first collaboration we initiated in Asia was a collaboration with an institute in central China, and we started this kind of collaboration in 1999. We put a focus on the capacity build-up, technology transfer, training of the involved scientists on site, and we also invited scientists to our sites in Switzerland. In 2006, the Chinese partner received from the Chinese government a significant financial grant to expand the implemented and established capacities in Wuhan. In the meantime, this group has its own research activities and other partnerships and business relationships established. And within the 10 years collaboration time, we experienced the isolation of one active principle, a compound which is today in early clinical trials. In 2002, we established a collaboration with the Shanghai Institute of Materia Medica. Here we put a focus on the isolation of plant metabolites originating from traditional Chinese medicine plants. We transferred again here significant analytical capacity to our partners, and that at that time the investments exceeded our in-house investments. 8 visiting scientists have been invited to our research laboratories in Switzerland, and we received from this kind of collaboration up to date 6,000 pure and structurally elucidated plant metabolites. In 2005, as Peter showed to you, we installed the partnership with the Biotech Research Institute, and I will show you in a second the key components from our perspective of this partnership with the Thailand Institute. In 2006, just to convey the complete picture to you, we installed a partnership with the Kunming Institute of Botany, also with the emphasis on the isolation of plant-derived natural products from traditional Chinese medicine plants. I would like to show you the key aspects of this partnership with the Biotech Research Institute. The contract was signed in 2005 between the CEO of Novartis at that time, Dr. Daniel Vasella, and the Thai government, just to have here a clear legal certainty that we are dealing with the right partners, because at that time it was not really sure whether the Biotech Institute would have been entitled to enter into negotiations with us. So we have been invited by the government of Thailand to enter in those kinds of negotiations, which have been signed mid of June. As you see from the figure, we are now in the 3rd term, which will hopefully be extended starting from 2014. But this is something very unusual. Normally, research collaborations are run for 3 years or 5 years, but that this collaboration holds on for 9 years now. This is really an exception in the pharmaceutical collaboration sector, I would like to say. The main objectives of the partnership was to support biotech to become a center of excellence in the natural products research arena in Southeast Asia, and also to include the Thai genetic resources into drug discovery efforts of Novartis. This is more or less a blueprint of a deal structure we used also in other parts of our collaborations with other partners. Novartis is responsible for the financial aspects, know-how transfer, and also taking care of royalties if we can generate any incomes from the genetic resources we are using here. On the other side, we are receiving samples from our friends in Thailand, and we are submitting these kinds of samples to HDS activities, screening activities in our organization. Biotech is responsible for the isolation of microorganisms and pure natural products, but I think it's important that you see that all these kinds of strains are not exclusive to our own research activities with Novartis. All these assets here around microorganisms are also be of benefit and use for the biotech research activities. So we don't have our exclusivity element here, and we may have a discussion on exclusivity and the need for that, because I have a little bit of different opinion than some other groups in that field. I think capacity building is one of a key element paired together with education. We are fostering and supporting the ambition of biotech to establish microbiology beyond of the fungal systems and using these kinds of biological resources for drug discovery and their own research activities at the Biotech Institute. And as pointed out by Gupta, we financed all the kinds of internships of the scientists and invited the scientists to the research laboratories in Basel. And at the moment we have one friend and colleague in the research laboratories of the Infectious Disease Unit in Emeryville in California to acquaint the Institute with the most recent endeavors and directions to screen for new antibiotics. This gives you somehow an impression what we have done in terms of educational events on site in Thailand. We have run at the very beginning various seminar series of Novartis experts at the Biotech Institute, and we have asked an internationally recognized scientific expert to implement actinomycetes related biology on site in Thailand. And this scientist is still around in that institute and is accompanying the direction of the institute in the actinomycetes related research. These are the four ladies which have entered our research laboratories at the very beginning. We have welcomed in Switzerland so far 8 biotech scientists, and they received training in chemistry, microbiology, high throughput screening, animal pathogen screening, and this all over the place is totaling in 23 months of education in Switzerland. I think this is a very important aspect of this kind of collaboration, that we are not just helicoptering in to our partners, but we are inviting the scientists, the young scientists of originating countries of, for example, Thailand, China, or now at the moment with Malaysia. We are inviting these kinds of scientists also to our research facilities and acquainting them with the newest technology. And Gupta pointed out, well, what we have in our research laboratories, the institutes cannot normally afford. But it's very important that the people know what is around if grants are opened up, so that they have a clear idea on the development in technology, so that these technologies might also be implemented at some time in our partner institutes in the originating countries. So what is the benefit of Novartis? We received in the 9 years of our partnership approximately 7,000 microorganisms. And I think it's important that you know that these microorganisms are owned by biotech, are owned by the respective countries where these microorganisms have been isolated. Novartis receives a time-limited exclusive user right, and we are supporting the research activities of biotech by giving the openness in this contract that these strains can also be used by our partners. In the meantime, when we started in 2006, 10% of all isolated natural products in my research group originated from the biotech collaboration. Since 2009, this is approximately between 30 and 50% of all our natural products originating from microorganisms there. However, also in this aspect of the collaboration, you see, patience is of imminent importance. We did not identify the path ahead by the identification of a drug development candidate. I think what was also mentioned as a key element of building trust is the transparency of the processes. And I very often hear in discussions, well, as soon as microorganisms are disappearing in organizations, you never can really find out what is done with these kinds of genetic sources. For that reason, we implemented a tracking system in our groups, which means that we link the strain with the originating country. So this is in a database which helps us to identify the link in this complex endeavor of drug discovery over many years from which areas the strains and extracts have been generated. We know at which day the extract was produced and at which day the respective strain has been populated in our research laboratories. So in the case we are isolating a natural product, we know exactly from which strain this natural product came and which the supplier was at that time. So with biotech it's very easy, but it reaches back to across the last 15 years. However, I would like to be also open to you. One problem in our organization are the old strains. The strains which have been isolated 30, 40 years ago, where we do not have appropriate documentations in-house. This is more or less, in our organization, the majority of the strains in my culture collection. With regard, which are the relevant aspects to build up a trustful and a successful collaboration around bioprospection, I think it's important that in the respective countries we have a national legislation with a clear regulation of access rights. And this is according also to the Article 6 of the Nagoya Protocol, as I learned from my IP colleagues in our organization. I think also very important and sometimes not clear in our negotiating partner countries is that the institutes needs an official entitlement of their governments to negotiate any collaborations around bioprospection. And this is also taken up in the Nagoya Protocol in the appropriate way. I think the inclusion of indigenous groups by the collaboration partner and governments are now officially part of the discussion, which is really helpful to reach legal certainty. I mentioned before the exclusivity and my view on that. I don't see really the need for an exclusive access to biological resources of a country as a necessary element of a collaboration. We at Novartis, we are absolutely free in that. We ask only for a certain time limit exclusivity to have the time to explore the potential of a certain scientific finding. But this is more or less 1 or 2 years, and that's it. But only on selected strains. All the other strains where we say, well, we received them, these strains are open to our partners to work with the strains and also to enter into collaborations with third parties without asking us anymore. I think the transferability of biological material to the laboratories, which is in some discussions also a sensitive point, as I learned, is an important part to maximize the likelihood of success. It's not about that the industry is not willing to transfer certain assays, but just the number of assays we have the chance to explore extracts, for example, inside of the Novartis organization is giving us a much higher chance to find a meaningful start of drug discovery and development activity. With regard to the prior informed consent, I think it's important that we understand that natural products research or drug discovery in general is a very complex endeavor. And sometimes when we identify, for example, let's take animal health as an interesting starting point of a compound, it might end up maybe in human medicine and not in animal health. So from my perspective, it's important that prior informed consent is defined in a broader meaning of the sense. So no focusing on a specific research area. And you may ask me why. And I think it's understandable that if we have to go back always in all the cases where we say, well, the prior informed consent was maybe the investigation of a certain compound in infectious diseases, but we see the potential in cancer. If we had to do this in my group of all these kinds of cases, we would not do research anymore. We would send out emails and writing letters all the time. I think this is something which is sensitive, I know, in the discussion, but it's also sensitive in respect to administrative burdens in a research group. With regard to access and benefit sharing, as pointed out also before, I see there is a clear need for an open and flexible negotiation chance according to the needs of the institutes. The institutes we are talking about are not always the same. We have very, very well developed institutes, as for example the Biotech Institute, but African institutes don't have these chances to reach these kinds of technological levels. This means that access and benefit sharing is a different component of discussions in these two parts of contract negotiations. I think also an important aspect is the equitable sharing of benefits, and this should be correlated to balance mid, short and long-term benefits, taking into consideration the risks and success rates. And we think that Article 5 here gives the opportunity to have or to access this region's kind of flexibility. Education, you have seen this before, is a key element of all our partnerships with the research institutes. And what was pointed out is the lack of knowledge how to use the biological resources is very often the driving force to approach, for example, mining. It's not necessarily in the Chinese and in the Thai related discussions the financial aspect. It's just how can we leverage the genetic resources in our country in a more meaningful as we have done this before. And I think sustainability with regard to education is a very important aspect. And money does not generate sustainability. Money is coming and going. But the knowledge base, the focus on knowledge base is a very important component in all partnerships I experience personally. IP and financial compensations, I think this is a no-brainer. And I think we have here good chances at hand to balance royalties versus milestone payments in a meaningful and a responsible way between the various contracting parties. With regards to ownership rights of the inventions, these rights, even if, for example, biotech would not be an inventor of a patent, does not mean that biotech does not take part in arising benefits from leveraging or using this kind of patent. The ownership rights are always defined in a contract. And as you perfectly know, and you may know it better than I know, the US-American patent law gives us not a chance even to use our internal contributors to be authors of those kinds of patents if they did not contribute to an inventive step. And I think this is a critical element here. However, we are taking care of them by a clear definition of ownership rights of the arising patents. And with that, I would like to close my speech. Thank you so much.